Class is class. Class time is class time, right? So we should start. Okay. So can you see the? Can you see me? The screen?
Yes, sir. I can see you and the screen both.
Okay, as well as the screen. So
Yes.
this is the best thing I have ever met by any single person. Everything should be made as simple as possible, but not simpler. Albert Einstein, you know him, he took life very lightly, but as well as very seriously. And you, this is very much pertinent to our understanding of neuroanatomy. That if you skip something that is needed, you can't actually get the whole circuit in a form. So you'll miss and you'll get to a wrong diagnosis. That's why neuroanatomy, study of neuroanatomy is a bit different than others. Say, for example, what I tell you, there are 7 relations of one, or the attachments, 7 attachments in a bone. If you forget 1 or 2, the teachers will not mind, right? But if there are 4 connections of a neuron and or a circuit, and if you miss one, might be you'll never be able to reach the correct diagnosis. And not only that, you will reach such a wrong diagnosis that would be harmful for the patient if you go for treatment on that basis. That's why in neuroanatomy study, everything should be known at least to its simplest form, but don't try to make it simpler. Okay. So these are what? Can you tell me?
It's a neuron.
And these two? These two are neuroglia, right? Glia also. So we are going to discuss today, we are going to discuss the neuron and neuroglia. And you can understand these are the very beginning, are very basic things of nervous system, as the neuron is the, before going to that, can I ask you some questions? Do the cells divide? Of course, right? If the cells were not dividing, how could we get so much population of cells all the time? Next question. Do neurons divide? Who can answer, please? Hello? Can you get me?
No, sir.
Hello?
Traditionally, they said that neurons
So
don't
neurons
divide.
are not divided.
Neurons divide in this early stage. Immature neurons can divide, but mature neurons usually don't divide. Other than some cells in the hippocampus and olfactory neurons, most all the neurons are, when they get matured, they can't divide anymore, right? So what is the utility of this? What could have happened if the neurons were dividing? What could have happened?
The size of brain would be enormous.
Enormous. So what? You'd be an enormously genius if you have more and more neurons. That's not a problem. Yes, that is also a problem. Why? Because you couldn't be even genius. if the neurons are dividing. This is called fleeting memory. In some clinical cases or conditions, this happens, that if the neuron starts to divide, what will happen? Say neuron A and neuron B, when joined together, it makes the letter P. P. The letter P is formed by when the neuron A and B meets. And now if the A is divided, B is divided, and now you try to mix up and match it, will you ever be able to make up the P again? Never ever. So there would be no memory at all, let alone being a genius. No one could have any memory if the neurons were dividing. No one could have any memory, any understanding, anything if the neurons are dividing. Right? This is not the muscle. This is not the bone that needs a single neuron, if it is fed properly and given the nourishment and environment properly, it can survive lifelong. So long I can live with my neurons. That's why the neurons are so important, that they won't be dividing, right? So this is what is the definition of the neuron then? It is the structural and functional unit of the nervous system which is responsible for reception, integration, and onward transmission of coded impulse. You see the coded impulse. That means a neuron will receive some impulse, it will integrate within itself the new impulse, and then it will transmit it onwards for the next neuron or next structure to meet, right? So this is, in short, very useful definition of neuron. And now if we see altogether what are the parts of the neuron, usually there are two parts. One is the cell body or soma, which contains the nucleus, and other parts, and one other are the processes or neurites. Together we call neurites. And neurites are again two types. One is axon, in Bengali axon, so he is or it is only one, and there are some properties of the axon, and other one is the dendrites. So dendrites must be plural, can be plural, and there are some properties attributed to the axon and some properties attributed to the dendrites. What are the properties? You can see axon originates from a specific part of the cell body, that is called axon hillock. And only from this part usually the neuron arises, and it is slender, non-branched, and even it branches at the end, and it doesn't taper. That means its diameter is all the way same, whereas the dendrites are tapering, shorter, and the branching pattern is there. Even I'll show you that even in these branches there are some dendritic spines. So, and they can develop from anywhere from the cell body. That is the extension of the cell membrane that is forming the dendrites, and only from the axon hillock region the axon arises. There may be, and there's a wrong understanding then, if there is only one process, we call it axon. No. And there are some neurons where there might be no axon, but still, if it is present, it will have these properties. And usually the dendrites carry the information towards the cell body, and the axons carry the information away from the cell body, right? So this is very important thing to note, that the dendrites will be carrying information towards the cell body, and the axon will be carrying the information away from the cell body, right? Okay. So you can see this is a magnified view, and where you can see the dendritic spines. These spines are clearly seen on the dendrites. Can you see this? The spines, the spike-like things. Can you see on the dendrites? So these are the dendritic spines. We'll be discussing about the dendrites and dendritic spines. And you can also see that this is the axon, and this is some sort of myelination that I'm not going to tell you today. When the nerve fibers will be dealt by other teachers, or maybe I don't know who will be, but that zone may be myelinated or non-myelinated. Depending on that, will be classifying that accordingly. That is a different issue. But let us concentrate on the axon itself. And one very important thing is that initial segment of the axon. What is this? This initial segment of the axon is which arises from a conical that is called the axon hillock. And after that, there's initial segment is the first 50 to 100 micrometer of axon after it leaves the axon hillock. So this is the axon hillock region, the conical part from where the axon arises, and this initial part is devoid of myelination. And why it is because it is the most excitable part of the axon where the action potential originates. So this is why this part is axon is this part, right? And normally, under normal conditions, the axon potential doesn't originate on the plasma membrane. That is, doesn't originate elsewhere on the soma. Instead, always the initial segment is so important to know. And sometimes it is frequently asked, not sometimes, it is frequently asked by the teachers. Now, some terms that we must know: sensory and motor, afferent and efferent. Sensory and motor and afferent and efferent, these are the terms that are very commonly used but very wrongly used ones, rather should I say. I'll try to make it clear by an animation. You can see it. This is the cell body of the first order neuron. So this one is carrying the information to the cell body. So this is called the afferent part. Now, these information is whether it is integrated here or not, it is going outside, and that is called efferent. But this was the cell body of the first order neuron. Now, this is the cell body of the second order neuron. But you can understand, this efferent neuron is no more efferent for this cell body because it is actually the afferent. It is carrying the information to the cell body. Again, this is again producing some propagation, and that one is the efferent. So you can understand it is not always that afferent is sensory and efferent is motor. Is it clear? We always say afferent is sensory and efferent is motor. It's not always true. In the same circuitry, one can change its role because these neurite or process is efferent for cell first order neuron, but afferent for the same process is afferent for the second order neuron. Is it clear? May I ask again, is it clear?
Yes, sir.
Yes. Why it is so important to know? Because you'll study it in details. I'm not going to detail it. At least 3 neurons are needed to carry a sensation from periphery to the cerebral cortex. Say, for example, I'm pinching on my hand. This sensation requires at least 3 neurons to reach it to the cerebral cortex sensory area. Again, I'm moving my hand. That at least requires 3 neurons as well, again, to get the command from the cerebral cortex toward, that is the primary motor area, towards the muscle, that is the effector area, or a gland, whatever. So at least 3 neurons are needed in a motor pathway, at least 3 neurons are needed in a sensory pathway. So all the 3 neurons are actually sensory neurons. Depending on whether it is afferent or efferent, that doesn't matter. If they're in a sensory chain, all of them are sensory neurons. If it is a motor chain, all of them are motor neurons. But as you can see, the same process of the neuron can act as efferent for the first order and afferent for the second order, right? So it's very easy. Now it's easy, but it is very tough if you don't think in that way. We are used to think that afferents are sensory, efferents are motor. Not always true, right? Okay, let's go to the next concept. Structure of the neuron. And you have always seen these type of figures to make it clear and simple, but it's not that simple. A neuron cell body is surrounded by so many other neurons processes, and it actually is in a such complicated state that a neuron has to survive or work. It's not this simple. But why we choose this one to draw? Because it is easy to draw and easy to understand, right? But we must think neuron is not alone. It is always working with group of other neurons. Okay, now let us come to the parts of the neurons. First of all, this is the plasma membrane of the neuron, and that is very much similar to the plasma membrane of other neurons. And you know, in the plasma membrane there is a cell membrane, and on the top of the cell membrane is a structure called glycoprotein, glycocalyx. A glycocalyx and glycoproteins all together with the cell membrane forms an barrier which doesn't allow semi-permeable barriers, rather to say, which allows some substance to enter and some are inhibited by that. So this is in short about the plasma membrane. We need not to detail it here. And cytoplasm again consists of the other cells, nucleus and cytoplasm. So this is the nucleus, and this is part of the, usually the nucleus remains in the center, and same, there are nuclear membrane, nuclear pores, chromatins, nucleolus, nucleoplasm, and you can see that here the nuclear, I mean there is no need to division of the cells, so they don't need to concentrate or condense or coil to form the chromosomes. That's why they are always in genetically active state and rather in uncoiled state. That's why you can see there is no deep colored chromatids here. All are sparsed, but the nucleus is very prominent because it is involved with continuous production of proteins. Because though, what do you think? The neurons will require protein more or less. What do you think? Because neurons don't contract, neurons don't secrete like muscles, they don't contract, but still neurons needs a huge amount of protein. Why? Because these proteins will be deliberated by the axons to the terminals, and they would be working as the neurotransmitters. So neurons requires a huge amount of protein. That's why the nucleus is very prominent in the neurons. Okay. So this is about the nucleus of the neurons. Next, cytoplasm. In the cytoplasm is the mitochondria. You can see there's plenty of mitochondria. The same reason why there will be plenty of mitochondria in the neuron, because it has to produce energy, or else even just think of that, even when we're sleeping, our skeletal muscles are almost inactive state. But what about the neurons? They are always acting. That's why there is need of huge amount of energy and supply of oxygen and glucose all the time. And there is a very limited time, say for example 1 to 3 minutes. After that, if the nerve, that is the oxygen supply or the glucose supply, is hampered to the neurons, these neurons will permanently be damaged, and this is very fatal, very much fatal situation. Understand? Okay, so this is the location and position of the mitochondria everywhere in the cell body. Next is the endoplasmic reticulum. As in other cells, there are two types of smooth and rough endoplasmic reticulum, and rough here is continuous with the nuclear membrane, you know it, for the same purpose. But one thing is important here, and that is very much important to know, because it would be asked by any teacher if a neuron is discussed or asked for. That is the Nissl substance. What are these things? These are the stacked upon each other. The rough endoplasmic reticulum stacked upon each other makes a pile, and these are known and Nissl substance or dense bodies. They are present all over the cell, but except this axon hillock. As you have seen the previous picture, the axon hillock is free of, I've shown you. Okay, fine, you know it. Axon hillock, I've shown you that the axon hillock is free of neurotransmitter, Nissl bodies. But they are present in the dendrites, but never in the axons. From the axon hillock down to the axon, there is no Nissl substance, but they can be present, as you can see, into the dendrites. Okay. Can you hear me? Can you see?
Yes, sir.
Okay. Now the neurophilum, I'm not going to detail it. You can see there are some parallel neurofilaments. These are neurofibrils. When they are broken into, they form the neurofilaments, right? And these neurofilaments and neurofibrils actually makes the skeleton of the neuron and shape of this neuron. Next is what? Microfilaments. Microfilaments and microtubules together, I'm not going to detail it because I don't have that much time. They actually maintain the propagation of neurotransmitters through the axons by forming some pumps, pumps, rapid pumps, small, slow pumps. I'm not going to detail it either. Just have an idea that they are actually for the transmission of the neurotransmitters and also for the formation of the processes, newer processes, and formation and retainment of the older processes. They are actually working together, microfilaments and microtubules. So neurofibrils maintaining the shape of the cell, and microfilaments and microtubules forming the newer processes and passage of the neurotransmitter along with these processes, like the axon, particularly in the axon. And what important thing to know that most of the cytoplasm of a neuron lies within the axon. That's why when an axon is cut, the most of the cytoplasm is lost. Understand? So I'm not going to detail it either. Next, the lysosome. There are three types of lysosome as well: primary lysosome, secondary lysosome, and residual bodies. You know it. Centrioles, that I want to highlight. Centrioles are, are the part of the cell which forms the site, and that is the mitotic spindles for cell division. So it is most likely that the centrioles will not be present here, but no, it's wrong. It's very tricky question. And all the time teachers ask, are the centrioles present in the neurons, mature neurons? You must have to say that yes, they are present, but they don't form the nitrous spindle, and neither they take part in the cell division. Rather, they actually maintain these microtubules and microfilaments. Understand? So here the role of centriole is not to form the mitotic spindles for cell division. Rather, they maintain the microtubules and microfilaments for the transport of what I have told, neurotransmitters, right? And lastly, some pigments are there, lipofuscin and melanin. And melanin is mostly present in substantia nigra of the midbrain. You'll study them in details. And so they have some specific functions. I'm not going to detail it any further. Okay, so in short, this is the structure of the neuron. And one thing is chromatolysis, that is sometimes comes as short note. Actually, what happens due to any reason when the Nissel's bodies goes to the periphery, it seems that they have been lost. And this, though they are present, they when they clump around the periphery, this phenomenon is called chromatolysis. Under stressful conditions, this may happen. This happens, not may, this happens that the Nissl substances goes to the periphery and it seems that they have been disappeared and been lost, and that is why it is called chromatolysis. But under favorable condition, they again come back and take up the whole, they take up their places in the whole soma or cell body, right? So this is chromatolysis, one very important thing. Now, classification of the neurons. According to the number of neuron processes, we can divide in multipolar, bipolar, unipolar, and pseudo-unipolar. So multipolar will have obviously an axon and multiples of dendrites, where the bipolar would have a dendrite and an axon. That is two. And this is the question, which one should be dendrite, which one should be, because both of them are looking same. But that answer you'll get from this one. I'm coming to that. And some are that where this is a peripheral process and that takes it is actually pseudo-unipolar, takes whether it can go to the cell body or it can bypass the cell body and go to the central processes. And these are called pseudo-unipolar. And right? So multipolar, bipolar, and pseudo-unipolar. now let's see. If we divide a single neuron into three zones or three regions: receptive region, conducting region, and the secretory region. You can see this whole part is receptive region, including the axon hillock and the whole part of the soma, and all the dendrites are receptive. And the axon itself is the conducting region, and where the axon terminates and forms some synaptic bulbs or synaptic structures, that is called the secretory region. But here you can see this trigger region reaches up to the cell body. So all this part is receptor, and this is the transmitter, and this part is the releasing or secretory region. And where here, this is the difference between these two. These two looks very much similar, but you can see here the coded impulse has to pass through the cell body. There is no other way than passing through the cell body in bipolar neurons. But in the pseudounipolar neurons, this can bypass the cell body or can go to the cell body, right? So this is the receptive region or trigger zone, and this is the conducting zone, and this is the releasing zone. This figure I have taken from Marieb's. Very excellent. It's an excellent book for understanding of anatomy and physiology. Marieb's Anatomy and Physiology. If you can collect it from it, I just got this part, not the whole book. That's why. Still, I can support you by providing these figures. This is a very nice book of anatomy and physiology altogether. This is Marieb's Anatomy. A bit precious book, about 16,000 taka its price, but still it's a very nice book. Anyway, next, relative abundance. On the basis of the abundance on location on human body, you can see the most abundant are the multipolar, and this is Purkinje cell. This is though in Bangladesh we call it Purkinje cell, but the pronunciation is Purkinje cell. Anyway, Purkinje cell or Purkinje cell, whatever you say. And this is pyramidal cell. This is present in the cerebellar cortex, and this is present in the 2 or 4 layers of the cerebral cortex. And these are multipolar neurons, and most abundant type is that. And rare, very much rare, is only in some spaces and regions. These are the 2: olfactory, mucosa, and eye and ear. These contains the bipolar cell. And unipolar, I mean, that is the, and they are only found in the multipolars, are mostly found in the central nervous system. And unipolar, truly speaking, the pseudounipolar are found mostly in the peripheral nervous system, and they are present in the dorsal root ganglia and sensory ganglia of the cranial nerves. You know the dorsal root ganglia. You will know, eventually you will know in the next classes, you'll know the sensory ganglia of the cranial nerves. But just remember, they are present in the ganglia, be it spinal cord, dorsal root ganglia, or the cranial nerves, sensory ganglia. They are actually present, these pseudounipolar. So what about the true unipolar? Who can tell? What are the true unipolar neurons? Can anyone tell me? True unipolar neurons? Okay, I'm giving you the answer. Write it down, please. There are 2 examples of true unipolar. One is the developing neuroblasts. Developing neuroblasts. Second example is the mesencephalic nucleus of the trigeminal nerve. You just write it down. Eventually you'll know these things. So one is the, what is this? Developing neuroblasts is the example of true unipolar. And another one is the mesencephalic nucleus of the trigeminal nerve. This is another example of the true unipolar. Rest unipolars are actually pseudounipolars. Okay. Next come, this is very much familiar one, lower motor neuron, you can see, and the shape, according to their shape, these some are stellate, some are granular, some are fusiform, some are pyramidal, some are having this sort of arrangement. So on the basis of appearance, we can say pyramidal, fusiform, stellate, or granular. So this is another classification of the neurons. But according to the direction of the impulse conducted, they are actually, you can see that the multipolar neurons carry the information from the CNS mostly to the muscles, and the CD-nucleolars carry the information from the skin or periphery to the central nervous system. And the bipolar neurons actually receive and send it to the central nervous system. So these in short about and from the understanding. So what do the, and that might be interneurons, that might be motor neurons, whatever, actually they carry the information from the central nervous system to the muscles. And the CD-nucleolars actually carry the information from the periphery to the central nervous system. And bipolars receive the information from the periphery and send it to the central nervous system. Both the side they are doing. Right. So this is on the basis, this is also taken from the Merriam and details. And this is the summary. And what I tell you, please study the Snell's neuroanatomy in details along with Visram Singh. That is the basic 2 books you need to know. And please try to concentrate on the charts given, provided in each chapter of the Snell's neuroanatomy book. That is enough. So rich these charts are and so summarized. If you just go through these charts, you will be able to understand at least 50% to more of the total chapter. So please try to go through the charts provided in Snell's neuroanatomy. Right. Okay, now this is one chart taken, and I have not yet mentioned one is type variety, that is Golgi type 1 and Golgi type 2. How much time do we have? How much time do we have? কটা বাজে?
12:30.
আর হাফ অ্যান আওয়ার পাবো না? ওকে
জি স্যার।
ওকে, আমি একটু দ্রুত বলছি। Golgi type 1 and Golgi type 2. Golgi type 1 is a long single exon. Long single exon, like the previous one. Here you can see this is a long single exon. So this is Golgi type 1 neuron. The Purkinje cell, the pyramidal cells, and lower motor neurons in the spinal cord all are Golgi type 1 because they have got some long neurons, exons. But these stellate in the cerebellum or granular cells, the cerebellar cortex, they don't have that kind of long exons. That's why they are called Golgi type 2. And other than that, interneurons are also Golgi type 2, which don't have long processes. And usually these Golgi type 2 neurons are inhibitory in nature. Usually the Golgi type 2 neurons are inhibitory in nature. And Golgi type 1 are excitatory, except this one. This one is a very exceptional cell, the Purkinje cell or Purkinje cell, whatever you say. This is though a Golgi type 1, this is an inhibitory one. Other than these two are very much excitatory, and these two, as the classification denotes, these are inhibitory in nature. So all the Golgi type 2s mostly are, or not all, mostly all the Golgi type 2s are inhibitory, and Golgi type 1s are excitatory, except the Purkinje cell, which is a strong example, very prominent example of inhibitory cells, being Golgi type 1. Okay. So how long a neuron can be? It can be 1 meter long, even more. A neuron can be 1 meter long or even more. I'm not going to detail it, find it out how. You will find it out. Okay. Now comes to the point, synapse. Synapse, you can see a picture where you can see there are some 2 neurons joining. I mean, they are coming in close contact, but there is a gap in between. So what is this synapse? Synapse is a junction that this part we miss. We usually complete it here. That synapse is a junction between 2 neurons. No, it's not always true. Or from 1 neuron to the effector cell, such as muscle or gland. That can also be, and that is also a synapse. So synapse is a junction that mediates information transfer from 1 neuron to the next neuron, or from 1 neuron to an effector cell, such as muscle or gland. This is the complete definition of the neuron. And now, what are the parts of the synapse? The parts of the synapse, before going to that, we can classify the types of the synapse according to structure is chemical synapse and electrical synapse. Chemical synapse will be detailing because that is very much evident, pertinent to us. Electrical synapse are not present in our us, that is human being. They are present in lower animals, and they actually has got two-way directional flow. That means very simple thing. There is a touches. In electrical synapse there is touch between two neurons, and there is a gap junction which actually allows the direct transfer of ions and electrical properties. That's why this is electrical synapse, and that is bidirectional. But in case of chemical synapse, this one is an example of chemical synapse, and we're going to detail it. It has got a axon terminal, synaptic cleft, and dendritic spine, or dendrite or axon, whatever. The, this is called presynaptic neuron, and this is the postsynaptic neuron, and there is a gap. And what you can see, these are the synaptic vesicles, and there are some, though it has not been shown here, there's plenty of mitochondria here to supply energy for these synaptic vesicles to bind with. And you can see these are the voltage gated calcium channels. So if the calcium is not bound to these channels, the synaptic vesicles will not open up, and the synaptic content, that is the neurotransmitters, are contained within these synaptic vesicles. One thing I've missed, the function of the Golgi apparatus, I've not written there. Actually, the Golgi apparatus are also plenty in the neurons, and these Golgi apparatus performs the usual function. They forms the synaptic vesicles, and the vesicles which contains the neurotransmitters and are directed downwards. That is in one way, and there is another. They can go backwards. That is called antegrade and retrograde passage of the neurotransmitters. And when they reach the synaptic terminal and the voltage gated channels are opened by the binding of calcium, then these synaptic vesicles fuses with the presynaptic membrane and opens up, and it has to pass a synaptic cleft. There is a cleft, and there are some microfilaments here that actually form a very velvety structure in between, velvet-like soft structure in between these two membranes. And these neurotransmitters actually binds with those velvety structures. And there are some enzymes as well in these velvety structures, which can destroy and reuptake of these enzymes are used. You'll be studying it in details in physiology classes. I'm not going to detail it. Now let us come to the postsynaptic membrane, where there are some receptors to catch up those neurotransmitters, and accordingly the excitatory postsynaptic potential, EPSP, or inhibitory postsynaptic potentials will be produced. Right. So this is in short about the parts of a synapse. Now, two terms related to these, neurotransmitters and neuromodulators. You can see from the previous picture, there are some neurotransmitters. So these are what? These are the main components, and usually one synapse acts through a single primary or principal neurotransmitter, say acetylcholine, noradrenaline. These are the principal neurotransmitters. So neurotransmitters are those which has a very short and rapid action on the postsynaptic receptors. But there are some other substances which may be present within the same synaptic vesicle or in different synaptic vesicles. Those are called neuromodulators. What do? They are also released in the synaptic cleft, and but instead of binding to the postsynaptic receptors, they actually enhance. They do four things. They actually act. They actually, what do they do? Have any idea? Neurotransmitters and neuromodulators. They enhance, prolong, inhibit, or limit the effect of the principal neurotransmitters. So they are actually not binding with the postsynaptic neuroreceptors. Rather, they are enhancing, limiting, prolonging, and inhibiting the function of the neurotransmitters on the postsynaptic membrane. through the second messenger system, G protein system, right? So by that, these are called neuromodulators. So there is a difference between neurotransmitters and neuromodulators. This is also very frequently asked question. You must know these things as well. So these are the some few terms regarding the synapse. Now let us come to the. I've taken these figures from Professor Manjara Shamim sir. He is the chairman of Bangabandhu Sheikh Mujib Medical University, and you'll see how the same concept can be converted by using an or application of some innovative ideas, should I rather say. And you would be proud to know that he is also a DMCN. He was a student of K34. So you are the students who can do such things, I believe, and you can see what changes or what modification he can bring to understanding. See the thing, types of synapse on the location of the postsynaptic neurons. This may be exodendritic. That means an axon is dendrite is being attaching to the axon, exodendritic. Exosomatic, so axon is going to the soma. Here is axon, and this is dendrite, so exodendritic, exosomatic, exoaxonic, and dendrodendritic that has not been shown here. Dendrodendritic, that means one dendron to another dendron. When they synapse, these are according to the location of the postsynaptic neuron. But just think about this. Will you ever forget this thing? If you can imagine that we have a shorter hand, a longer hand. So this is the dendrite and this is the axon. So this is exodendritic, this is exoaxonic, and this is exosomatic. So this is how you can change your ideas forever with the application of some innovation, right? Okay. So what are the properties of the synapse? One-way direction in chemical synapse, particularly because I have already told you the electrical synapse has got two-way direction process, exchange of two ways, but in chemical synapse it is only one way, right? Why one way? Only one way because you see in the I don't want to go there. You have seen that in the postsynaptic membrane there is no synaptic vesicles, so through which it can come back again to the surface. So it is only one way direction, chemical synapse. And synaptic delay, what does it mean? Okay, let me go to that, because without going to that I'll not be able to tell you those things. So you have seen there's presynaptic vesicles in the presynaptic membrane. That's why it can come up here and be released. But what about this one? It can't go back because there is no postsynaptic vesicles there. That's why this is one-way direction. Synaptic delay, that means these neurotransmitters requires a very precise, but it requires a time to reach the postsynaptic. Can you understand me? Are you with me? My students, are you with me?
Yes, sir.
Can you understand, please? Can you understand? I'm confused. You're not talking or making any sound or any response. That's why. Okay. Please raise your hand or let me know if you don't understand. Right. This is the problem of lecture with Zoom, that I can't see you. Okay. So when these has to cross this barrier, that's why it requires a time. This is called synaptic delay. Third thing is fatigue. That means if all the postsynaptic receptors are blocked, and so long they are being attached to these neurons, no more neurotransmitter can bind to these postsynaptic receptors. That's why this is called fatigue, that this has to finish up their work, and then next group of neurotransmitters can bind up. That's why this is called neuronal fatigue or synaptic fatigue, right? The last properties that I need to tell you with the figures, you can see feed forward inhibition and feed backward inhibition. These two you must know, and I'm detailing it in detail a little of these. The feed forward inhibition, you can see this is the first order neuron, these two are the second order neurons, and you can see these first order neurons are excitatory for the second order neurons, and these two second order neurons are also excitatory. But what happens, it has got a collateral branch and there is another inhibitory neuron in the pathway which inhibits, actually, which when it is stimulated it actually inhibits because it's an inhibitory, the second order neuron on the right side, but the left one is stimulated all the way through. Feed forward inhibition, though it is forwarding but it is actually inhibiting this one, the second, the right hand side second order neuron, but the left hand side second order neuron is getting stimulation all the way through. Why it is too important to know? This is for important for knowing that, you see, when we say that there is a common rule, if a agonist or prime mover has to contract, there should be equal relaxation of the antagonist, right? That is called law of reciprocal innervation. You have studied it. I have told it in earlier classes. The law of reciprocal innervation, where if the prime mover, say for example, I want to make contraction at the elbow joint, I have to have the contraction of the brachialis, biceps brachii, coracobrachialis. At the same time, I have to have the equal amount of relaxation of the triceps, or else there won't be any contraction, right? So how does it happen? See, this, say for example, this is the upper motor neuron, and these are two lower motor neurons. One is for the agonist and this one is for the antagonist. What happens? The upper motor neuron is stimulating, there is contraction. At the same time, an inhibitory interneuron is being stimulated by the collaterals, and these inhibitory is actually inhibiting the antagonist muscles. So you got this. This is called feed forward inhibition. Next, feed backward inhibition, where the same cell, this is Renshaw cell, you will study. I'm not going to detail it. You will study it eventually in the proper time and proper maturity. Now it's too much for you, way too much for you. So I'm not going to just have an idea. Renshaw cell. And this Renshaw cell is an example of feed backward inhibition. I'm not going to detail it any further. So these are two inhibitions, feed forward inhibition, feed backward inhibition. Now, one more thing regarding this neuronal pole. You will frequently hear these terms: divergence, convergence, serial processing, parallel processing, and reverberation. Divergence, you can see one neuron is first order is neuroning 2, second order is increasing, so this is called divergence. And convergence means all four, they are converging to one. Serial, parallel, you can understand, and this is called reverberation. Okay, so these are terms you'll, particularly these two, you will face many times: divergence and convergence. Okay, now the neuroglia. Can you spare me 5 more minutes? Do we have 5 more minutes? কয় মিনিট বাকি আছে? 5 মিনিট?
Yes, sir.
ও আছে তো, অনেক সময় আছে, 15 মিনিটের মতোই তো আমাদের আছে না? ওকে, ঠিক আছে, আমরা 15 মিনিটে শেষ করে ফেলতে পারবো। নিউরোগ্লিয়া ইনশাআল্লাহ শেষ হয়ে যাবে। Neuroglia, previously it was considered as nerve glues. That means just scaffolding. They were thought that they would be filling up the blanks between the neurons. They have no role. It was originally thought. That's why they have given the name nerve glue or neuroglia. But now it is proven that they have got plenty of functions. Right, so they are no more nerve glues only. So 4 types of the neuroglia are present in the central nervous system, and 2 types are present in the peripheral nervous system, right? So these are the neuroglia present in the central nervous system: astrocytes, epididymal cells, microglial cells, oligodendrocytes. These are the 4 types of neuroglial cells that are present within the central nervous system. And these 2 are the neuroglia present in the satellite cells, which surrounds the cell body of the neuron. and the Schwann cells. Schwann cells are also, these two cells are the neuroglia present in the peripheral nervous system. Before going to the details of these, just I want to make it clear and very distinctly know, you must know that the neuroglia are plenty. It outnumbers by 10 times. That is, for maximally, for one neuron, there might be 10 neuroglial cells, though they are very small in size in comparison to the neurons. They are very small in size, but they are plenty, outnumbers by 10, 10 is to 1, right? So this is very important information regarding the neuroglial cells. Let's start with the astrocytes. Astrocytes are again two types, protoplasmic and fibrous astrocytes. The protoplasmic astrocytes are found in the gray matter of the nervous tissue, and the fibrous astrocytes are found in the white matter of the nervous tissue. Do you know what is gray matter and what is white matter? Of course you know. What is gray matter and what is white matter? Can anyone tell me in details? Or anyway, what is gray matter and what is white matter? Gray matter,
স্যার গ্রে ম্যাটার হচ্ছে সেল বডিজ গুলো নিয়ে
right?
যেটা
আর কি বলবা? Cell bodies, glial cell, you must tell these. That's why the glial cells, the cell bodies of the neurons, the glial cells, and the blood vessels. আরো ভালো বলতে চাইলে, and also the initial part of the axon, though it's not cell body, but the initial parts of the axon are also included in the gray matter. যাই হোক, ওইটা বলতে না চাইলেও হবে, আন্ডারগ্রেডে না বললেও হবে. You didn't need to tell. At least you have to say the cell bodies of the neurons, the blood
Yes.
vessels, and the glial cells. And what matter? What is white matter?
হোয়াইট ম্যাটার হচ্ছে
Processes.
Axon,
Process of the neurons. Then same thing. The glial cells and blood vessels. Right? এটা কিন্তু বলতে হবে. ওই দুটো বাদ দিয়ে বললে হবে না. If you just say the cell bodies of the neurons are gray matter, no, it's not correct. The cell bodies of the neurons,
গ্লিয়াল সেল এন্ড ব্লাড ভেসেল
glial cells and blood cells. And white matter, cell body processes of the neurons, collection.
গ্লিয়াল সেলস
And
এন্ড ব্লাড ভেসেলস
That's right. Okay. So I've told you that there are two types of astrocyte, and one is called the protoplasmic astrocyte. What we can see here, they are present in the gray matter and white fibrous. That's why white fiber is always white almost. That's why you can make easy understanding and remember it easily that fibrous astrocytes are present in the white matter of the central nervous system or nervous tissue. Now, what are the function of these astrocytes? There are 12 to 13 functions of the astrocytes. 12 to 13 functions. Of them, you must know some important ones, like they form the structure. I have not written it here because it is very clearly written in your book, the Dotto, even in the কি বলে ওটাকে? Visram Singh, or even in snails, it is written. That's why I couldn't get the time to write it here actually. But what I want to show you that how do they form the astrocytes? Astrocytes actually, they form the supporting framework, they form the scaffolding for the migration of the immature neurons, they form the electric insulator for the neurons, they form barrier for selective passage of substances, supports by providing nutrition selectively, and also they maintain the potassium balance within the central nervous system or neurons. So they have got some astrocytes may act as phagocytic cells. So there's plenty of functions of astrocytes. It might be a short note written on astrocyte. But what I want to make sure that you understand, this is blood-brain barrier. I'm not going to detail it, you'll study it but just see the blood-brain barrier. Part of it is formed by the astrocytes footplate of astrocytes form the blood-brain barrier, which maintains the selective barrier between the blood and the brain tissue. So this is called the blood-brain barrier. So you can, from this figure, you can remember, at least you can remember that the astrocytes food plates form the blood-brain barrier. That's enough for now. When you will study in details of blood-brain barrier, there will be layers of blood-brain barrier, where the blood-brain barriers are absent. All these things you must know. What is the purpose of blood-brain barrier? You must know these things. Without knowing, you can't actually pass, because this is very important for fetal purpose. Say, for example, which drug can be given during intrauterine life that will not be affecting the central nervous system. If a drug can cross the blood-brain barrier, that will hamper the fetus, even within the mother's womb. So this is very important to know the blood-brain barrier. Next, ependymal cells. This is also a neuroglial cell present in the central nervous system, and this is the brain or spinal cord tissue. Then it has got some ciliary process on the fluid filled cavity, and this is the inner aspect towards the nervous tissue. And what it does, actually, it passes the cerebrospinal fluid is passing through. From this figure, you can see the cerebrospinal fluid is passing through these cells, and they actually help in circulation of cerebrospinal fluid. And these ependymal cells are again divided into three categories. Ependymal cells are ependymocyte, tannocyte, and choroidal ependymal cells. There's a list in your book of snails. You can study it from there. Choroidal ependymocytes actually involved in circulation of cerebrospinal fluid and absorption of cerebrospinal fluid. The tannocyte is another one of ependymal variety, which has got long processes, and they can sense the chemical substances within the cerebrospinal fluid, and accordingly they transfer the substances from the cerebrospinal fluid to the hypothalamo-hypophyseal system. Hypothalamo-hypophyseal portal system they can transfer. So this is a bit tough for you to understand right at this moment, but just have an idea that there are some cells which are called tannocytes, which can read up, which can smell the chemical composition, and accordingly they transfer the substances. And choroidal ependymal cells, the third variety of ependymal cells, they actually secrete and produce and secrete this one is the choroidal, which can produce and secrete the cerebrospinal fluid. So this is in short about the ependymal cells. Now, microglial cells. These are actually the variant of the mononuclear phagocytic system prevailing all over the body, such as in the skin they are called Langerhans cells. In the tissue they are called histiocyte. In the liver they are called Kupffer cells. Similarly, in the central nervous system they are called microglial cells. They remain inactive so long there is no inflammation, but when there time of inflammation, they act as defensive cell in the central nervous system. And oligodendrocyte is another microglial cell which have processes and form sheaths around the myelin. Myelin sheath within the central nervous system is formed by the oligodendrocytes, right? So these are in short about the four categories of glial cells present in the central nervous system. Now come to the neuroglial cells in the peripheral nervous system. As I've told you, these are some neuroglial cells which surround the cell body of the neuron, and they are called satellite cells. Just it is an imagination that this is the earth and these are the moons. That's why they are called satellite. These name came from it's an analogy, very occurred analogy, but still it is workable one to remember that they would be around the cell bodies of the axons. And one important thing is the Schwann cells. Schwann cells actually form the myelination in the peripheral nervous system. So I've told you the central nervous system myelination is formed by the oligodendrocytes, and the peripheral nervous system myelination is formed by the Schwann cells. But there is a huge difference between the myelination process by the Schwann cells in the peripheral nervous system and the oligodendrocytes in the central nervous system. But I don't have the chance to discuss it here. When the nerve fibers will be discussed and the process of myelination will be discussed, then you can understand. And you must remember what is the difference between myelination by the oligodendrocytes in the central nervous system and the myelination by the Schwann cells in the peripheral nervous system. That is a very important thing to know. Okay, now come to this figure. This is taken from your snail's anatomy, and each in single figure tells and shows everything: the plasmic astrocytes, the neuron, the fibrous astrocytes, the epididymal cells, the capillaries, the blood-brain barrier, the oligodendrocytes forming the myelination, and the microglial cells are there also. And this is another protoplasmic astrocyte, and they are forming the blood-brain barrier here. Blood-brain barrier are formed by that. So this one figure tells and shows many things, and the relationship between the neurons, the blood vessels, and the glial cells. Right. So this is another chart taken from Merriam's. You can take it from the snails as well. Last thing I want to show you, very important one: plasticity. Plasticity of the neuronal circuits. Neuronal circuits are not fixed. Say, for example, there was a trauma to the brain or pathological damage to the brain. If the brain lesion is not large, what happens? There is functional recovery commonly occurs. You have seen. Have you seen it? Even the tracts are cut. Sometimes it happens that when the nerve is cut, sometimes for a few days there is no work. Again, there is recovery by physiotherapists. If the physiotherapists continue the physiotherapy, and the functionality is at least to some extent regained. So that is what happens there. Say, for example, recovery from paralysis or caused by stroke is commonly seen. You can see in clinical practice, right? Haven't you seen that a person was paralyzed after stroke and the physiotherapy is going on, and after a few days he or she can get the full functionality back or partially back? How it happens? So this is called plasticity. Very important thing to know, that functional recovery involves the taking over of the functions of damaged neurons by neurons that remain intact. এটাই কি বলা হয় plasticity. The reorganization of the connections within the brain is known as plasticity. When if a part or the neurons in a part of the brain is damaged, sometimes it is not, if it is not a very wide damage or very serious damage, the functionality of those neurons can be taken up by other neurons. Again, another example of plasticity is that learning of often repeated tasks. Say, for example, a surgeon doesn't need to think about how much deep I should cut, how should I peel up, how should I retract the structures. It is very common day practice. Say, for example, neurosurgeon. For a neurosurgeon, cutting the brain is like cutting a cake, right? He or she is doing it, walking, thinking, dancing. I know one surgeon, Professor T. Chaudhary. The more serious, the more complicated the surgery, the more joyful he becomes and starts to sing in the OT. When he starts singing, others understand that he is enjoying the surgery, and the more complicated it is. So this is the present adaptability used for learning of often repeated tasks. This is also plasticity, right? So plasticity is one when the neurons take up the function of other neurons, be it pathological condition, be it in normal condition, and this is called plasticity of the neurons. And here is actually structural changes occurring. That is very important to know. It doesn't happen simply that they are taking. Actually, the structural functional relationship is changed. That is called plasticity. That neurons change their structure. I've told you the structures are maintained by neurofilaments and neurofibrils, microfilament, microtubules. So they take their change in their shape and their arrangement, and all the organelles are changed, and the neuron itself accordingly changes its function and takes up the function of other neurons. That is called plasticity. Okay, with this, I hope you would be able to understand the basics, very basics. There are so many things I couldn't touch because of shortage of time. within 1 hour. Actually, I can't go further. And you, for the general purpose, actually you don't need any more. But if you are interested, you can go through the Merriam's Anatomy. That is a very nice book. But you must have to go through the Snell's New Anatomy first, with the help of the Vishram Singhs. Okay, so with this, I want to conclude today's class. Thank you very much. Any more question, please? Any question, please? Okay, in that case, we can stop. Okay, ক্লাস শেষ করি আজকে মতিন, তাজ, শশী, যাদেরকে দেখতে পাচ্ছি।
ওকে স্যার।
ওকে, ভালো থাকবেন।
আসসালামু আলাইকুম স্যার।
ওয়ালাইকুম আসসালাম।